Well, hello. I am going to call this file Insight 1. And it's because of some of the mistakes that I saw people make on the first practical. I'd like to take a look at this practical, which I gave in July of 2017 to help you understand how I put it together. Okay. First and foremost, when I'm putting a practical together, I always have the lab packet with me. Lab packet. And of course the problem with this is I don't necessarily see the information. That's not spelled right. Maybe it is. Okay. I don't necessarily see the information the same way that you see it. And what I mean by that is maybe I'm too familiar with it, or maybe I see some piece of detail that you didn't see because you just took a picture of the model and you haven't actually looked at it closely. Or you haven't asked yourself, what does it mean to look at this model and what are all of the pieces of information? So this should make sense. Look, you've seen this. I'll say identify, and then I'll tell you what it is. And this is a spinal cord section. And you'll get, what, a minute and a half at the station? But it shouldn't matter if I gave you 30 seconds or if I gave you an hour, right? Hour would just mean I could ask you more detailed questions. A minute and a half means I can ask you some pretty serious questions about all of the basic structures that you should know about the spinal cord, the afferent information traveling in, and the efferent information traveling out. And you'll notice that I picked this model for a reason. I mean, it has all the vocabulary on there so that, number one, you can identify what's going on. So this is, if you're looking at the patient, this is the right posterior gray horn. Right? And now if I ask what is it, I'm asking for right posterior gray horn. Most people will just give me gray horn. You'll get some credit for that. But, you're looking for that level of detail at this point in your career, right? You're always thinking, okay, where am I in relation to this model? I know I'm missing the bones that are supposed to be here. I know I'm missing probably most of the meninges. The dura and arachnoid are definitely missing, but chances are the P is around it. And you'll notice that this is pretty straightforward. It's almost too simple, right? And in doing so, what's going to happen, of course, is you're not going to take it seriously. Number two is looking at that space here, okay? And that space, of course, is the anterior median fissure, okay? And notice I'm not pointing to the white commissure or the gray commissure here. The end of the needle is what you're looking for, okay? Makes sense? Good. So I've just called time, and now you've moved on to the next model, and the next model is 3 and 4, and, looks like you can see number 4 here, and if you can imagine, you're looking at this entire model, you're looking at the left-hand side. Did I say right or left? It's the right-hand side. Whatever. I might have actually gotten the previous thing wrong. Sorry about that. You're looking at the left-hand brachial plexus, right? Okay, and I know that because I can see the cervical plexus coming off here, and here is our phrenic nerve, right? Phrenic nerve, P. Phrenic nerve, okay? And that's coming off of our cervical plexus. So chances are I have some other structure that you should see on this really large model. And in reality, if I had 10 minutes to give you on each of these models, I could put 10 minutes worth of identifiers on it. But remember, all I'm doing is looking at those structures that are on your lab packet. Okay, and by lab packet, I mean those first 2 sheets that we say, look, this is what you need to know for the practical. Now, here's a similar question to the first one, where I'm looking now posteriorly to the actual structure, right? And I know that because here is the body of the bone and here are the bifids. And this is the model of that cervical structure inside the body. Okay, and you can see here that here's our dura mater and our arachnoid mater. And I'm pointing into this space. And I'm not looking at what's over here, and I'm not looking at what's back here. You need to know that this is the subarachnoid space. And chances are, if you can give me subarachnoid space, you can give me cerebral spinal fluid. Okay? So if that's what I'm looking for there, what I'm looking for here is remarkably easy. This is that dorsal root ganglia, and that dorsal root ganglia, when you look over here, you can't make it out. Okay? So if I put an arrow on this, like I did over here, I could take, literally, took that arrow and I put it over here, okay, I would probably take dura matter for that, right? You'd give me, it's the dorsal root ganglia, but the actual dorsal root ganglia is what's beneath it. And then because the reason why I would look for dura matter, of course, is look, there's the dura matter, there's the arachnoid matter, and here's the actual subarachnoid space. Now here's a model that you have to do some thinking for, right? And that is, if you've done your reading inside the, your textbook and inside chapter 12, you know that this is let's draw a multipolar neuron for you here, right? Yay, quick sketch of a multipolar neuron. Yay, axon terminal. Yay, and let's make it a myelinated axon axon for you. So it all makes sense, right? We'll put some dendrites on it for you. Woo! Cell body, okay? And inside class, you're going to see I'm going to put more and more information in this as we're going along. But at this point in time inside the lab, you should have looked at this model and go, okay, what are these little yellow dots? And these little yellow dots are, of course, vesicles. And we know vesicles are made from the Golgi apparatus. And those vesicles should have something inside of them. And that something inside of them, of course, is neurotransmitter, right? So NT. And inside class, we're like, oh, we got to learn what those different NTs are. Great. It's like lab, we never learned what they were, right? You just learned that they're neurotransmitters. Pay attention to that. You're also going to see here that I have this needle pointing to something right there. Well, what is this? Well, this is the actual membrane of the cell. And because it's an axon, this is the axolemma. So that's learning the definition of what axolemma is and applying it are two different things. The number of people who get this type of question wrong is insane because they don't stop and go, oh, okay, this is a cell membrane of an axon. Axon is an axon lemma, okay? Just like every cell membrane is. Plasmal lemma, okay? Again, this is a question, identify the structure at the end of the needle. And people come up to the stationist, they look at it and go, WTF. And then some people will pause and they'll go, oh, I know what this is. Oh, look, they've resected the brain out. They left some bones in there. And he's pointing to a piece of the brain. Okay? And if we know this is the spinal column, we know inside that is the spinal cord, we're probably looking at some part of either the pons and or the medulla. Okay? But you probably want to think about why I would pick a model like this. Well, because it has pieces of the information, and I'm looking to see if you can think on your feet while you're looking at that list. Remember, it's one thing to memorize the list. It's another thing to be able to apply that information. Here's another example, right? So I've taken the head off of a model, and remember your last practical, you probably had to look into this at some point in time. And, well, now I need you to apply the information again. Okay, well, inside here, 13 and 14, well, this is a specific structure inside the brain. I'd love to tell you what it is, okay? It'd make your life easier. It'd certainly make my life easier to make sure everybody inside the room knows that's the Tuatara Gland, right? Yes, no, maybe. But over here, people aren't expecting this. They're not expecting to be looking at the cerebellum cut mid-sagittally so you can actually see the white structures inside there. And inside your lab packet, that's simply called arbor vitae, right? But on a practical, I'm going to look for a way for you to see it that you're not expecting to see. Now, every lab has some set of questions that are what I consider to be remarkably obvious, right? So you're going to see here that this is questions 17 through 20, I think, right? But all you can actually see here is 18 and 19. And you're going to notice the end of the needle is actually pointing inside a space here. And I want you to think about what that is. I could tell you what it is, but you'll notice now I'm giving less and less information. This large white structure here, though, I'll tell you what that is. That's the corpus callosum, right? That's the axons of neurons that are now communicating between the right and left hemispheres of the brain, okay? But it takes a little bit of ingenuity to figure out what that is. So maybe you go try and figure out what that is. So by now you've figured out that even if you don't believe me, the best way to get information into your brain and communicate to another person is to actually be able to draw and explain what you're doing, right? So question 21 through 25, turn over your practical and draw and write out the components to the reflex arc. And we only will go over this quite rapidly inside lab because from our perspective now, this should be a pretty trivial idea to you. But using the colors we see inside the textbook, let's go blue. We're going to take some piece of sensory input, switch this over to purple, where we then send into an interneuron, and then we're going to have, because it's a reflex, some decision has been made that you don't have to think about at all. We're now going to have some sort of effector. And now what we want to do is we want to make sure that you know that there are 5 parts to this, right? So there's going to be some sensory receptor, SR. That's going to travel along some afferent neuron, AN. That afferent neuron is myelinated. And in fact, this is our unipolar neuron whose cell bodies are found inside the dorsal root ganglia, DRG. It synapses with an unmyelinated interneuron, which is where the thinking happens. This is literally the control center, CC, but it's an interneuron. I'm just going to put int underneath it here, where it's like, okay, what pathway do I need to travel on to make sure you understand how to move quickly? You'll see how we use this inside lab. Now, this signal is then sent to our efferent motor neuron. Okay, so I'm just put EN here, where it's going to release some neurotransmitter to an effector. And that effector is then going to make sure that some result happens, right? So sensory receptor is 1, afferent neuron is 2, interneuron is 3, efferent neuron is 4, and then finally the effector is 5. So there are 5 parts to our reflex arc. Doesn't this look familiar? Okay, you know what it is. You notice where all the needles are pointing. Notice why this is so important. Okay, if these are somatic sensory receptors inside the body, these are unique somatic sensory receptors found inside the skin. Now you're going to see, at least in this example, we're about to finish here, that what cranial nerve innervates this structure. So in lab, you do have to learn the 12 cranial nerves, their name, their number, that sort of stuff. Where do they come from? But remember, I ask that you understand where the nuclei are of each of these cranial nerves. So understanding that the vagus nerve has the nuclei inside the medulla means that when you read that part of what its function is, you learn how it actually regulates not only the heart itself, but it can regulate other structures. Another example here is, what cranial nerve cell bodies would I find in this structure? So here's that model of the brain. And just to finish up, you should be able to look at this and go, I can convert this into a really quick cartoon that if I have to, I can think my way through. What cranial nerves would I find here? What cranial nerves would I find here? What cranial nerves would I find here? Or maybe, what cranial nerves would I find out here? Wah, 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 wah. Let me give you an eye. Dink. Dink. Whack. Or how about down here when you're smelling? Okay, that's enough. Hopefully you have a better idea of how I'm going to design this practical so that it's, this is the nose, by the way, in case you weren't able to figure that out. Okay? How I'm going to design this practical exam so you can be excelling. Okay? Have a great night. Take care. Bye.